Okay, I'm just kidding. All right, so chapter number 17, we're going to learn different types of tools when it comes to diagnosing infections. So first thing we need to do, if you have the role of a physician, you're working in a facility, is try and figure out what is causing the infection. Okay? And there's a number of different ways that we can go about this. Really, what you ideally want to do is you want to pinpoint it down to the level of genus and species. Yes, knowing it's a staph infection is beneficial, but knowing what type of staph is even more beneficial. So is it a Staphylococcus aureus infection, or is it a Staphylococcus saprophyticus infection? Like, what type of staph is it? That is absolutely ideal, getting it all the way down to the level of genus and species. Okay, so we need to know how to properly sample from a patient and then which tests to run. So that's what we're going to talk about today. A few different categories for tests. First up, phenotypic just relates to what something looks like. So if you were to grow the bacteria on a plate, what do the colonies look like? Are they large? Are they small? Are they kind of star-shaped? Are they yellow? Like, what do they physically look like? Also, under the microscope, what shape is the bacteria? Okay, if you see a Diplococcus, that can eliminate a few types of organisms, of course. Physiology is, basically what that organism does, what metabolic processes, it utilizes. It's biochemistry. Most of the time you're testing if the bacteria has a certain enzyme. For example, oxidase test, catalase test that we did with lab 41. Does your bacteria have the oxidase enzyme? Let's run this test and find out. Those are phenotypic biochemistry type tests. Everything on this intro tube right here, these are all biochemical tests. These are all phenotypic. You're basing your answers off of that, color change based on these metabolic processes of these organisms. Immunologic, this is when we're going to be using antibody-antigen interactions. Okay, so you're going to obtain, serum sample from a patient, run some tests. Okay, so you're going to use the patient's blood for that. Genetic tests, this is when we're going to be utilizing DNA, RNA, different types of probing. Western blot, for example. Okay, hybridization type techniques. Genetic testing is a really kind of, I would say, the most reliable type of testing to do. Okay, because it's based off the genetic information, which there are databases where these organisms have been sequenced, and we can reference them. Okay, they're always working on lots of new techniques as well, new technologies. Here we can see various locations where we can collect a specimen and how to do that. One important thing to remember is practicing aseptic technique all of the time. Okay, so you need to practice aseptic technique when you're obtaining samples. You do not want to contaminate these samples, of course. Okay? So you want to try and isolate that infectious agent and not introduce anything else into that sample that shouldn't be there. So you can see, capturing some saliva sample, a swab. So if you're taking a swab from the back of the throat, you don't want to get a good sample, from the back of the throat, let's say, and then run it along the top of the patient's tongue. Okay, well, that's going to contaminate it. There's all kinds of bacteria in the mouth. You want to get exactly, that one location. Sputum samples, nasopharynx swab. I think Kaylee actually talked about that when she went through her MRSA presentation. Of course, blood. Urine, so taking a catheter or clean catch. Okay, so here you'd be using an antiseptic, on the external portion of the urethra, and then the patient would urinate, and then you catch it midway through. You see, from the skin, you can obtain a swab around the surface of the skin, or you can use a scalpel and get a little bit of a deeper sample, more of a tissue sample. Some CSF fluid from a lumbar puncture spinal tap, okay, to see what may be present in the CSF. Fecal sample, vaginal sample, skin's there. Next up in here, I can't remember, I think actually Kaylee showed this too. Looking at a culturette, okay, And it's this really nice type of a transport medium, a transport device, where you can take this to the patient. Here's that sterile swab. Obtain your sample, put it back into this culturette. There's some media here. The media doesn't do what media does in lab for us. It's not going to facilitate growth of the organism. You're not trying to grow more of it. You're just trying to remain, keep the organism viable, not kill it. Okay, keep numbers exactly the same, not allow it to die, basically. Okay, so you can safely transport it back to the lab. Okay, so different steps when it comes to these different techniques. So first thing, of course, you need to talk to the patient. You need to have that, physical examination with the patient and discuss signs and symptoms. It's always the first thing which is going to take place. And I do have a little chart here that shows this. Okay, so first thing is you're starting with that patient analysis. Of course, you talk to the patient, see what's going on. So clinical signs and symptoms, see if any of those match up right off the bat. What is an in vivo test? What does in vivo even mean?
In vain? Usually in life.
There's in vitro and there's in vivo. Vivo means inside the person, inside the living host. Okay. Where in vitro, that would be run outside of the body. Think like in vitro fertilization, for example. Okay, so run in a test tube, run in a well plate, okay, run on an external source. In vivo, the most common example of an in vivo test, okay, would be the tuberculin test for tuberculosis. Okay, because you're running that test within the patient's body and looking for a response on the patient. That's in vivo. And then running immunological, serological, so on the serum, the blood of the patient. Okay, this is all starting with the patient. Let's go back here. Okay, so you're looking at mucus production, okay, taking some samples. Second step, okay, let's analyze what we collected. Okay, so we have these collected and we're going to analyze them. Direct tests, okay, pretty straightforward. Looking at them underneath the microscope, running a gram stain, for example. Seeing what we have going on. Okay, running antibody, antigen type tests. Okay, so immunological type tests, like what we're going to do in lab next week, the ELISA. We're going to do an indirect ELISA. We're trying to detect if there's any antibodies in the patient's blood for HIV, in the patient's body for HIV. Let's go back to this little table. Okay, so now you have the actual organisms. You can run some of these direct tests on them. We'll talk about a few of these today. Macroscopic, okay, so here, a lot of these, the difference between these two, macroscopic type tests, you can see the results with the naked eye. Microscopic, of course, you would need to utilize a microscope in order to do this. And sometimes a basic microscope, like what we have in class, or a more high-tech microscope where the results would be on a computer screen. Culture isolation, again, we'll talk about a few of these today, but one that we've done, antimicrobial sensitivity, Kirby-Bauer. Okay, so we'll see which antibiotics are useful against that particular organism. Okay, so you'll run the different tests on what you isolated from the patient. And when all is said and done, you have some presumptive data, which is a general idea. It could be like the level of the genus, perhaps. It's not very specific, but you have a general idea of the type of organism. Confirmatory data, that's really what you want, because that can get down to the level of genus and species. Yes, it is a staph aureus infection. Yes, it is a MRSA infection. You know exactly what it is. That's ideal. Does it always happen? No, but it's ideal. Okay, so again, presumptive, you're getting down to the genus level, and then confirmatory, hopefully you're getting down to that species level. So looking at how quickly some bacteria can grow, and also different times for tests to have their results. If you were going for a rapid strep test, how long do those results take?
5 minutes.
5 minutes. Okay, it's gonna take a few minutes, not very long. If you took a sputum sample and, we're trying to grow some bacteria on a plate and you're looking for tuberculosis colonies, they take weeks to show up. That's going to take a very, very long time. That's a very slow growing bacteria on this artificial media. Okay, so depending on what you're doing, results can be fast or, it can take a little bit of time. Again, sending strep away for a culture, okay, that's going to take a couple of days. Think about when you put bacteria on a plate in here. You don't get the results by the time you're done with lab, right? You sit in the incubator for a couple of days. And the next time you have lab, you see the results. Sometimes it can take longer. All right, so looking at this, so if you're looking at, let's look at athlete's foot first. Okay, athlete's foot, you don't really need to run any tests when it comes to athlete's foot. Okay, so just looking at, the patient's signs and symptoms that they are presenting, what they're experiencing. Okay, you can safely diagnose athlete's foot. Okay, so looking at the AIDS virus, looking at HIV, okay, the virus responsible for AIDS, isolation of that actual virus from the patient is very difficult to do. So instead, what you do, of course, you're looking at signs and symptoms, but you're doing a serological test. You're running a test with that patient's blood. What are you looking for? You're not looking for the virus, you're not looking for the agent, but instead you're looking for the patient's response to that virus. Does the patient have antibodies against HIV? That's what you're looking for. Okay. So depending on what you're testing for, there's a lot of different tabs that you can go. This is a pretty nifty little table. This picture's right out of your book. Okay, so this is what a physician would send off to the lab. Okay, so date, time, person collecting. Here we can see the source of specimen. Okay, so what do we have? We have a clean catch urine sample, sputum sample, cervix, stool sample, whatever you might have here. What type of test do you want run? Okay, and then the lab will fill out the rest. Okay, so here, is it gram-positive cocci, is it gram-negative cocci? We're going to write in some results. Over here, culture results, how many of these types of organisms were recovered from that sample. Okay, so, an enteric organism, they find E. coli, Enterobacter, what did they find? And again, how much, based on their scale. And then very interesting down here, this is results for Kirby-Bauer. So what we did in class with the antibiotics. Okay, so here we can see, they're going to be resistant, intermediate, sensitive, and then you can come up with a good, antibiotic plan based on this. That's pretty cool. Let's do a concept check. We got a bunch of true, false questions. All right, so the first one, saliva is sterile and is valuable in sputum collection. That's very, very false. Both parts of the statement are false. Okay, so it's definitely not valuable in sputum collection, and it is not sterile by any means. It's a ridiculous amount of microorganisms associated with the saliva. All right, true or false? Careful antisepsis is needed when taking samples from sterile materials such as blood and CSF. That's true. Okay, you don't want to introduce anything into those samples. All right, next up, true, false. It can take several weeks to isolate and identify microbes in tuberculosis infection. True. For sure. That bacteria grows super, super slow. It can take a really long time if you're trying to grow that up on a plate. All right, true, false. Some diseases can be diagnosed based solely on presenting signs and symptoms. True. Yeah, some can. Not all by any means, but some definitely can. And so athlete's foot, for example. All right, let's talk more about these phenotypic methods. We're going to go into all the categories and talk about the genetic ones, immunologic ones, and of course the phenotypic ones. A lot of the phenotypic ones are things that we perform in class. So up there on the screen, we can see results here from a gram stain. So the purple, is that gram positive or gram negative?
Positive.
Positive, for sure. So the pink would be gram negative. According to this picture, it's a little blurry, but purple looks to be what shape? Cocci, and the pink appears to have a rod shape. Again, it's a little blurry, but That's what it appears with that picture. Okay, so running different types of stains. And staining is really nice because the results are fast. You know, think about how long it takes you to do a gram stain. Yeah, it's not the fastest type of staining, but really it takes 5 minutes. Okay? It doesn't take a very long time. The longest part is waiting for that smear to air dry, which of course you can be doing other things at that time. Okay, isolation media. So here's the thing with isolation media. Think about lab 6 that we did in here where you took a sample of an object, or maybe you left the lid off. That was like one of the first labs we did this semester. Lots of things grew on your plates, right? Well, let's say you're taking a sample from a patient and you don't want lots of things to grow on the plate. You're just looking to see if they have this one particular organism. Does anybody remember the kind of media? This goes way back to chapter 3 in your textbook. The type of media which is only going to grow, you know, that one select microbe, just that one type, and prevent everything else from growing. Selective. Okay, so selective media. So you can select for one type. We did use a selective media in here. It was a Sabarod agar plates that's selected for mold only. Bacteria weren't able to grow on those mold plates that you took. So it's selective for mold. But you can get media selective for Enterococcus or selective for Escherichia coli, E. coli. So you can get it more selective. So if you're looking to see if they have an E. coli infection, nothing grows on that E. coli selective media, you can rule out E. coli. It's really nice because you don't have all the other organisms interfering. Okay, you're just trying to grow one. It's a really kind of cool media to do. Okay, so selecting the media is not a big part. You just don't want to choose anything. You need to see what, you know, your ideas of the organism can, you know, they can grow in. Okay, so if it's present in small numbers and easily overgrown, this is actually what happened when we did lab 9. Remember, you guys made the mixed cultures. Which bacteria of the 3 was the gigantic bully?
Serratia marcescens.
Serratia marcescens, that red bacteria. How much did you put into that tube?
One drop.
1 drop. That is it. And it took over the entire plate. The one that didn't want to grow was the Micrococcus luteus, and we put in a ton of that bacteria. We put in like 3 mils. We put in the most Micrococcus, and yet it was completely overtaken by Serratia. So again, if the pathogen is present but is in small numbers and easily overgrown, like if Micrococcus was a pathogen, doing what we did in lab is not going to work to see if Micrococcus is there because Serratia took over. So that's why you want to get, you know, this specialized, this selective media to kind of pull out just Micrococcus and prevent other things from taking over. So very, very important to really think about the media. So here, just a stat. So 80% of UTI is caused by E. coli. So using a selective media for E. coli would be, you know, a good means. Pure cultures is really what you want. Again, like I said, you don't want to have a variety of organisms when you're running a test. Think about this. Okay, so think about this entero tube. You took one plate and you put it at the end of a little needle, dragged it through, twisted it back, capped it up. Great. Trying to figure out what that one bacteria is. You don't want to take this from a sample and have like 5 bacteria in here because then the results are not going to work. So you want to make certain you have a pure culture. You want just one organism. So you might have to take a sample and you might see a few and then you may have to separate them, similar to what we did with lab 9. We're going to pick the red one and grow it on its own media, pick the yellow one and grow it on its own media, and so on. Because you can't run these tests. You can't run the oxidase test if you have like 7 different bacteria because you don't know which of those are positive or negative. But if you can separate those 7 bacteria and then run the oxidase test on each of the 7, then you can get more answers. Okay, so you want to have a pure culture. Lots of different tests we can do here. So we'll talk about a number of these as we go through. Motility, does anyone remember the type of media that you would use for motility? This one we talked about. We didn't physically. SIM. Yep. So It's a semi-solid type of media. Okay, it stood for sulfur indole motility, SIN media. Semi-solid, so you can kind of see the organism moving its path of where it moved around. What it looks like on a culture, different results for staining, oxygen requirements. We use the anaerobic jar, so we use that brewer's anaerobic agar. What media did we use for oxygen requirements? Remember it had the green on the top?
Was it FTM?
FTM.
Oh, yeah.
Yep, it was FTM. Fluid thioglycolate media. Okay, so FTM. And then you can figure out, is it an obligate aerobe? Is it an obligate anaerobe? Is it facultative? What are its oxygen requirements? And you can eliminate a number of bacteria. It turns out to be an obligate anaerobe, then you have a select few to choose from. Biochemical analysis, similar to these tubes that we'll check today. Antibiotic sensitivity test, Kirby-Bauer. Okay, seeing which drugs they are sensitive to. Here's yet another example where you can take a look at a gonorrhea infection, Neisseria gonorrhea. Okay, so looking at this particular infection, if you do, of course, if you look at, you need to have signs and symptoms in the patient. So if they're experiencing those symptoms and they have, pus from the genitalia, let's say, and you want to run a test on that pus. Okay, so here you can run that gram stain, of course, along with the signs and symptoms. And if it comes out gram negative and it's diplococci, that's gonorrhea bacteria. Okay, a patient should not have gram negative diplococci associated with the genitalia. Okay, so that, along with those signs and symptoms, that may be enough to say, hey, it's probably a gonorrhea infection. Would they run further tests maybe to confirm that? Probably. Wouldn't be a bad thing to do, but right off the bat, they can kind of safely assume that. Okay, but further tests would be, ideal. Okay, biochemical testing. Again, phenotypic. A lot of these tests, the results are going to be shown with color change. Example is this. What we did with these tubes. We did that oxidase test. Remember, we had the swabs. What exactly are we testing for? If I were to say, Anna, run an oxidase test on your bacteria, what is Anna looking to see about her bacteria? What is she trying to learn about her bacteria? What's the point? Is she trying to see what her bacteria does to oxidase? Like, what is oxidase? Is it a toxin? Is it a nutrient?
It's an enzyme.
It's an enzyme. Oxidase is an enzyme. Okay, so Anna would be checking to see if her bacteria has that enzyme. Does Anna's bacteria have the oxidase enzyme? If yes, results will turn out purple. If no, it stays a tannish, brownish color. Okay? And when we ran these tests, everyone had a negative result for oxidase. Let's hop back here real quick. This is kind of the general schematic of what happens. And again, we're looking at a color change. Most of these tests are based on enzymes. So here's what happens. You have your bacteria, you don't know what it is, so Anna doesn't know what it is. She has her substrate, which would be the oxidase reagent. If her bacteria has oxidase reagent, then you have a positive result, which would have been purple. If it doesn't, meaning oxidase is absent with her bacteria, then it doesn't change color and it's negative. Usually a color change is positive for a lot of the different tests. Another example would be that catalase test with bubbling. Okay, the evidence of bubbles, positive. No bubbles, negative. Okay, so catalase and oxidase, definitely two tests I want you guys to be familiar with, okay, for the next test and also for the lab practical course. Carbohydrate fermentation, this is one that, actually, let me draw some here. Let's draw something real quick up here. This is going to play a role with today, lab 41. Okay, so carbohydrate fermentation. What we're asking, so if I were to have you do a carbohydrate fermentation test, you would say, does my bacteria, okay, so your unknown bacteria, does my bacteria ferment this carbohydrate? Whatever the carbohydrate is. So for example, I asked you to do a glucose fermentation test. Does my bacteria ferment glucose? Or I could say run a lactose fermentation test, or a trailose, or a mannose, or a mannitol, whatever it might be. Sorbitol, whatever it is. Whatever. Okay, so you're looking to see if it ferments. What were products? Let's say this. Three things we saw as products resulting from fermentation. What's one of them?
Alcohol.
Alcohol.
Gas.
Gas, carbon dioxide gas.
Acid.
Acids. Which of these do you think would be the easiest to measure? Like, how can you tell if alcohol is produced? Taste it, smell it, right? We're not going to be doing any tasting, but I mean, what do we do in lab? We kind of smell it, right? Is that reliable, quantitative? No, it really isn't. What about gas? You can see that. You can see it bubbling. Okay. We used a balloon where it captured the gas. Okay, so that's possible. What about acids? Can you measure if acids were produced?
Yeah.
How do you do that? Test strip, pH test strip, right? So this really is the easiest way. Sometimes gas can also be utilized, so here's what we would do. And actually 5 tests in this strip are in this category. Okay, 5 chambers. The first one, and then these 4 in a row. These are all carbohydrate fermentation tests. We have glucose, inositol, lactose, arabinose, and sorbitol. 5 different carbohydrates. You're checking to see if your unknown bacteria can ferment them. Now, if we didn't have that very handy-dandy, convenient test strip, this is what we would have to do. We would have to get a tube. Okay, so within this tube, I'm going to use the color red because this is what it looks like, and this is what your strip looked like prior to inoculation. It was red. Why is it red? It has phenol red indicator in it, and it has your carbohydrate in it. So if this was the glucose fermentation test, it would have glucose in it. So it has a pH indicator already in it, and there's glucose in here. So what do you have to add to it? Your bacteria. Add you'll add your unknown bacteria. 2 different results may happen. Here's one. Here's 2. So in the first one, let's say stayed red. Yeah, you might see some cloudiness in there because the bacteria grew. That's fine. Okay, we're really not going to see that with ours in the strips, but if you use like a liquid media, it can kind of be throughout, like so. it stayed red. And the other one, we see yellow. A few dots in here. So you, of course, will have bacterial growth in a liquid media. Again, these test strips are not liquid. So now, this one is red, this one is yellow. Why the color change? Because of this. Phenol red indicator. It's a pH indicator. Instead of going back to chemistry and using like a pH meter or a little test strip that you dip in and measure the pH, it's already in there. All you have to do is look at the tube. If it stays red, red is neutral. Did fermentation happen if the pH stayed neutral? No. So we'll say no glucose fermentation. Yellow, yellow equals an acidic pH. So did fermentation happen if acids were produced? Yep. So this would be positive. That'd be positive for glucose fermentation. So looking at, this is Amanda's. So we look at Amanda's tube. Here's her glucose test, the first one. It's yellow. So is it positive or negative? Positive. If we look at her sorbitol, which is this one right here, it's nice bright red. That would be negative. Okay, so that's how you would check these results. Okay. All right, so that's taking a look at carbohydrate fermentation. To measure gas, in these test strips, they put wax over the top. If the wax gets pushed up, you know there's gas. Liquid, of course, the gas would bubble away. So sometimes what you can do is you can put a little mini test, I mean, they're literally this tall, like that wide, they're very, very tiny. We put a little test tube in here, upside down like that. And then if there's gas produced, gas bubbles, will go inside the test tube, and the test tube will float up a little bit, just so you can tell if there was gas. So put that in there. Called the Durham tube. Very, very tiny little. Okay, so I felt like that one was worth talking about because we'll see it with the 5 times. 5 of them are carbohydrate fermentation. So there's so many different tests you can run based on enzymes. Okay, so looking for enzymes is a really easy way to go. This is what we did. And again, think of how simple this was opposed to inoculating 15 different test tubes. You just put the bacteria through a strip. And again, looking at glucose, here we can see it turned yellow. That's going to be positive. Like Donatol, it stayed red, that's negative, and so on. When you check your results today, here's how you do it. Just a little mini preview. Anything positive, you circle the number underneath the test. If it's negative, you don't circle the number. And then the ones that are grouped together, you add up the circled ones. 2 plus 1 is 3. 2 plus nothing circled is 2, and so on. And then you get a little 5-digit code, and you look it up on the computer. So that's how we'll check our results. This test is not a carbohydrate test, so for this test, yellow would be negative. It's only the carbohydrate ones when yellow is positive. Here's the API 20E test strip. This is lab 40, where you'll need to reference in order to answer a particular question in the lab. So it's a little bit more difficult to do. You have to suspend the organism in a saline solution in order to deliver it to this strip. Okay, it's a little more involved. You end up with a 7-digit number opposed to a 5-digit number. Here's an example of a flowchart. I just expect you guys to be able to read these. Guaranteed test question. I might have on there, I might say, all right, you performed a gram stain and you ended up with a gram negative, or let's say a gram positive cocci. And then you ran the catalase test and it was negative. what's your bacteria? Okay, so you should be able to say, okay, it's cocci, it was gram-positive, it's catalase negative, streptococcus. Okay, so you don't have to memorize these results, you have it all right in front of you. This is what I got, A, B, and C, and hey, that's what it is. Okay, so you should be able to read a flowchart. Phage typing, pretty cool type of test you can do. Phage typing is something used most commonly when they're trying to figure out an epidemic. Oftentimes like a food type epidemic, like Salmonella or Campylobacter. Okay, so looking at Shigella, looking at those types of epidemics. What they do is very, very similar to Kirby-Bauer. First off, what is a bacterial phage? What is a bacterial phage? Talk about the virus chapter.
Oh, jeez.
So a bacterial phage is a virus that attacks bacteria. Remember, they looked like, I'll show you here, they look like this. Okay, little, like they're mechanical spider-like. They attach to the bacteria and they can destroy the bacteria or they can become integrated into their DNA. Okay, so this whole test is taken off of that, thought that there's these viruses that attack bacteria. So remember when you did Kirby-Bauer, what was the first thing that you did? Like, how did you get your bacteria on the plate? Did you do a quadrant streak? Did you do a zigzag? Like, what did you do?
We did a lawn.
You did a lawn. Like, you covered that whole plate with the bacteria. Do the same thing here. And then remember, you stamped it. Well, you stamped it with discs. What was in those discs?
Antibiotics.
Antibiotics. In this scenario, you start off the same way. Cover your plate with bacteria. Instead of stamping it with antibiotics, you stamp it with these viruses that attack bacteria. So if you know in like this first quadrant, this virus attacks this strain of E. coli, and it kills E. coli, okay, then we can see that our unknown bacteria would be E. coli. Okay. When you guys did your lawn, you knew what bacteria you're putting on the plate. When you do this, you don't know. Okay. Instead, you're seeing which viruses kill the bacteria, and then you can look up and say, okay, I know this virus kills this bacteria, and it did it on my plate. That's probably the bacteria I have. So here you have these different quadrants. So here you can see whatever bacteriophage this is in 52A, okay, it killed the bacteria. And then also number 79. So whichever bacteria is susceptible to 52A and 79, that's going to be the unknown bacteria. So kind of the same theory, but you're kind of working backwards. You're trying to figure out what an unknown is based on knowledge about which phages kill which bacteria. It's really cool. And then Kirby-Bauer, okay, we just discussed that. So again, you make your lawn of your culture, and then you stamp it with the drugs to see which drugs are effective. And again, when we did it, we knew what bacteria we put on, okay, with it, and then you can see, what works and what doesn't work. It will be best for the patient's treatment. At first glance, looking at this, you probably say, okay, whatever drug this is, it appeared to work the best. But again, remember we have standards. You have to look at the table and see which are sensitive, resistant, intermediate. Significance. So if I ask you a couple questions like, if you were taking a urine sample from a patient and you grew it up on a plate and you had a colony of E. coli show up, does that mean they have an E. coli infection? E. coli is normal flora. So if you see a colony, maybe 2 colonies, it's really not a big deal. What if you see hundreds of colonies? That would be a big deal. That would be a probable E. coli infection. But 1 or 2 really doesn't mean much because it is a normal flora. What about if you had a sputum sample from a patient and you had 1 or 2 colonies of tuberculosis? would that indicate probable infection, or do you think, oh, it's just normal flora? Probable infection, okay, mycobacterium tuberculosis is not normal flora, okay, so that's going to be, a big sign that's, you're looking at that probable tuberculosis infection. Where 1 or 2 E. coli, that's normal flora, that may be okay. Obviously, hundreds of them, not okay. So just because something grows, you need to think about what it is and how significant it is. Just something to keep in mind. Let's do a concept check, a couple, then I think I have 2 slides, one more concept check, and then we'll give you guys a little break. So this one, non-sterile specimens. That means we have multiple bacteria. They're often plated on what type of medium?
Selective.
Selective. Remember, we're trying to select for one, we're trying to pull one out. You want to have actually what's in, and we'll set it, you want to have what's in bullet point 2, which would be a blank culture. You need to have before you can run a biochemical test like this. Pure. You want to have a pure culture. You can't run a test like this, or your enterotube, or an oxidase test, or a catalase test, if you don't have just one bacteria. You can't run the Kirby-Bauer if there's 3 bacteria mixed together. You need to have a pure culture. Biochemical tests are often based on microbial expression of a particular enzyme. So we're looking for an enzyme most of the time, not always. Okay, so this one looked at, if it can ferment, whatever carbohydrate. Other ones like oxidase or catalase, okay, look for an enzyme. Blank typing is often used to trace bacterial strains in an epidemic.
Phage.
Those phage typing. Okay, when they use those bacterial phages. Okay, so yes, let's run through these 3. We'll do a concept check and then take a little break. All right, so next up we have PCR. How many of you guys came across the term PCR when you were doing some research? Okay, it's about half of you guys. PCR, it stands for polymerase chain reaction, and it is just that. You can take the smallest amount of that organism, the littlest, tiniest amount of genetic material, which may not be enough to run a series of tests. So it would be ideal if you had a lot more of it. So what a PCR does is it takes the smallest quantity and you can make millions of copies of it. And we have an abundance of material to run a series of tests with. Okay, so it basically just takes the smallest amount and multiplies it. Okay, so that's what PCR is. It's numerous copies of DNA or RNA genetic material within hours. Use a little thermocycling machine and can make lots and lots of copies. So you're amplifying what you have. Okay, and that can make a lot of tests run more efficiently. You're going to get better results. Okay, next thing I have is to take a look at hybridization. This can be a little confusing. So what I'm going to do is I'm going to draw something on the board. Right over here, I'm not going to pull up the screen because it's not going to be that much up on the board. So do you remember back in the, about halfway through the semester where we talked about complementary bases? So what binds to A?
T.
T, what binds to C?
G.
G, okay, it's based off of that concept. So what you would have is you would have something known as a probe. A probe is a known DNA sequence. You know what the sequence is. So let's say E. coli. You know if you were to look at a certain strain of E. coli, it has these bases in a row. I put 6 bases on the board. We're talking hundreds, thousands bases, base after base after base. That specific sequence is only found, those bases in that exact order, is only found in that strain of E. coli. That's your probe. Mine is very short up here because I want to write lots of bases. Okay, so I'm only using 6. So it's based off the premise that you know this strain is with E. coli. Now you have your unknown organism. Your unknown organism, let's say it's DNA, you end up finding this. TAAGCC. This is unknown organism number 1, and then you have unknown organism number 2, which happens to be TAACCG. Okay, so 2 different ones. Okay, so let's say, Amanda grabs a sample from a patient and she has the unknown, DNA. And she's like, I'm thinking it's the strain of E. coli, so she'll use the probe, which is complementary to, this strain with E. coli. And she takes it, and she's trying to see if there's a match. Okay, and with the probe, this is the probe, there is always some type of a fluorescent dye or fluorescent tag attached. This dye will glow when there is a match. So if Amanda takes her strain, her unknown bacteria, and she tries to match it up with E. coli, is that dye going to glow? A goes with T, right? T goes with A, T goes with A, G doesn't go with G. So hers is not going to glow. Okay, let's say Anna has an unknown organism. She's like, I really think it's that strain of E. coli. Okay, well, she's going to utilize that probe. And this one, if you look at it, this is a perfect match. So what's going to happen? The dye will glow. And she'll be like, all right, I know that this is what I have. A very reliable type of technique. We'll talk a little bit about specific tests you can run using this idea. But at this point, I want you to know this is probing, also known as hybridization, because they're hybridizing, attaching. It's all about this base pairing, these complementary bases. Okay, so this is complementary to this strain of E. coli, and then they're seeing if there's a match. So here. So here we take a gram, we have a patient sample, we run a gram stain, we see it's staph. We do our gram stain, we know it's gram positive, it's toxic, all right, we narrowed it down to staph. But what type of staph is it? So let's use this probe, which is complementary to Staphylococcus aureus. You want to know if it's Staphylococcus aureus. Here's a little dye, a little fluorescent molecule. Okay, so here's these complementary bases. We're going to run this situational hybridization. And then if we see this glowing, it's because there was attachment. Okay, so that would be positive results. So we know that this unknown bacteria is that strain of Staph aureus. Really cool techniques that we can run. All right, so to sum this up, microbial DNA and RNA, how can you rapidly amplify them?
PCR.
PCR. What does PCR stand for?
Polymerase chain reaction.
Polymerase chain reaction, exactly. And then fluorescently labeled probes are used in what type of technique?
Hybridization.
Hybridization techniques. Yes, those would be hybridization.